So as an early career scientist, it's extremely disheartening to know that 3 of the 4 global bleaching events have been in my lifetime. These bleaching events, local and global, are only predicted to increase in severity and frequency as warming continues. So it creates a special urgency to coming up with innovative restoration techniques that may help us maintain diversity of the GBR while we're mitigating climate change. So as someone who studies evolutionary biology, adaptation, and genetics, something that I could contribute to this was contributing to the base knowledge that might make marker-assisted breeding and selective breeding techniques beautifully explained by Kate and Carly and promoted and proposed by Lena Bay, Marlon Van Oppen, and Ruth Gates. So why do we even think this is possible? We think this is possible because of something called intraspecific variation. It's just a different response within a species to the exact same stimuli. So here in this image, you can see the same species of coral in the same environment, but there's a different bleaching response. There's variation in that bleaching response. And you can arrive at this difference in bleaching response biologically in a few different ways, but the literature tells us that genetics can also play a big role in this difference response. So that's what I wanted to look at. So what is the evidence needed for marker-assisted breeding? What kind of things would we need to see? So we might want to see that there's heritability. We want to know that increased thermal tolerance can be passed from parents to offspring. And we want to know that there's adaptive potential within a population. We also might want to find genetic markers that are associated with this thermal tolerance so that we can maybe pick out thermally tolerant individuals. But these markers have to reliably predict thermal tolerance, increased thermal tolerance in these individuals. So to gather that evidence, we've done experiments where we assess variation in survival given a heat stress, and then looked at heritability of survival in that same heat stress. So survival is our phenotype, that's what we're looking for. And then I do a SNP frequency analysis. So SNP stands for single nucleotide polymorphism. You can think of this as a single base pair change in a coding or non-coding gene. So genetics 101, there's your chromosome pair. At a specific region in the genome, you might find two different base pairs in a single individual. I'm going to look at that change in frequency in a large group of individuals from a control and from heat stress. So for all of these experiments, we need to know the parents. So we need to know the genetic background of these individuals. So we went to Esk Island Reef. We collected 6 rabid adult colonies, and we brought those into the CSIM. And when they spawned, we collected their gamete bundles. We separated eggs and sperm, and then made these very specific crosses. So if I could draw your attention to this chart over here. So each coral got a letter label. And then each shaded region gives us our cross. A, in this case, is dad. B, in this case, is mom. And that's consistent for every single cross. Dad first, mom second. And we use those terms to refer to specific families as we go forward. And then you get families of coral larvae. So here is a family. I did this for all 10 families, but this is an example family that I will show you. So we need to split these up now for these 3 different experiments. So for survival and heritability in survival, I need to be able to monitor individuals. So I put 20 individuals into each of 16 replicates, and then randomly distributed those across our control tanks and across the heat stress tanks. For our SNP frequency analysis, we need many more individuals. So we put 500 individuals into each of 4 replicates and randomly distributed those across controls and heat stress tanks. So then we get our data. This is very exciting. So this is a very standard Kaplan-Meier survival analysis. So on your y-axis, you have survival probability. On the x-axis, you have hours in a heat stress. So we go from DB, had family DB up at the top, has the least mortality, and family CA down at the bottom has the most mortality. So survival probability varies quite a bit within family. The difference between these corals is their genetic background, which means genetics are important for increased thermal tolerance. So DB, an individual in that group has a 75% survival probability after 240 hours in heat stress, whereas CA has a 75% probability of dying after 240 hours of heat stress. So now we know it varies. Great. But can be passed on. So now I have to do a different type of analysis. So this is a heritability calculation. This square term right here is a historical biological artifact. It means absolutely nothing. That term is not squared, so ignore it. What I would like to draw your attention to, though, is these V variables here. So VA is variation that is contributed by the genetics of the parents. And this variable also takes into account the genetic relationships between individuals in the study. VP, on the other hand, is total variation in this survival phenotype. This also includes the environmental component, so whatever variations contribute to the environment is going to be in that variable. So parental genetics, variation provided, attributed by parental genetics over gene variation is what we're calculating here. And then we end up with our heritability estimate. Heritability estimates range from 0 to 1, 0, no heritability, 1, all heritability. We have a heritability of 0.66. One of the higher heritabilities is actually in human height, and that is 0.8. So this is a pretty exciting value, because this 0.66 means that not only can increased thermal tolerance be passed from parents to offspring, but also there's adaptive potential in this population. And I'm going to illustrate that with another example. My colleagues, Nathan Kirk, and our collaborator, Emily Howells, went to the Persian Gulf. They have very, very thermally tolerant corals of the same species in the Persian Gulf. They did a heritability estimate, and it was 0.43. This 0.43 estimate means that there's actually less room for adaptation in that population because they have lived at those higher temperatures for many, 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 many years. So selection has already happened for them. So this number is very exciting. But onto our SNP frequency analysis. So to identify these genetic markers, I do this SNP frequency analysis, and we want to be able to, I'm going to talk about what we're comparing here. So in this survivorship distribution, in the control, we have all survivorship phenotypes, which means we have a full and complete SNP frequency distribution. So all SNPs are going to be here because no individuals have died. They haven't experienced any kind of heat stress. They're just fine. But then, when you go over to the survivorship distribution, post-heat stress, many individuals have died. So what we're sampling is the tail end of this distribution, and we're not going to have a complete SNP frequency distribution in this population because only individuals with alleles that confer thermal tolerance are going to make it through this heat stress. So now we can get to the data. So each family, this is just a logistic regression, determining if these SNPs are significant. So you can see the family name in the bottom corner of each graph, and each one of those black dots represents a SNP that we identified. So we identified 11,600 to about 24,000 SNPs in each family. The red dots represent SNPs that are changing in frequency after that heat stress. So these red dots represent SNPs that change, that have changed in abundance after we look at that thermal stress. So we identified 34 to 336 of these significant markers. So you identified markers. Cool. Super exciting. Where are they? Like, what do they do? Do they predict thermal tolerant individuals? So, I wanted to look at the significant SNPs and see if they overlapped between families. So I did a comparison. I did a lot of Venn diagrams. I did every three-way comparison. I'm only going to show you two of these comparisons. Oh, no. Sorry. No, back. Okay. So you can see all significant SNPs, and there's quite a bit of overlap between these two families, or between any of the two families for that matter. But when you look at the three-way comparison, you see that there's only three, or only two. And when I look at all the different Venn diagrams, that's about the average. That's what I see. So these 3 and 2 are very exciting. They're mostly found in non-coding DNA, and looking at these markers for validation for thermal tolerance is exciting, but we'll talk a little bit more about conclusions in a minute. The next thing I wanted to know was, are any of these genes in functional genes, are they functional proteins? So the columns I'd like you to pay attention to are the crosses. So here's all the families that these genes were found in. You can see that there's overlap in some. And then you want to pay attention to the descriptions here. And you'll notice that some of them are not annotated. That means that these are novel genes that we don't find in other eukaryotic organisms where we have much better and more complete genomic information. So as more genomic information for corals comes online, we'd expect to maybe find out what these are. But as of right now, we do not know. However, the ones that we do find are canonical heat stress genes that are found to be significant for increased thermal tolerance in other studies. So all those RNA-seq studies, these are the same genes that are found, which gives me confidence that what we're seeing is real, these markers are real. But you also notice that the overlap is not super great. So what does this mean for marker-assisted breeding in a restoration context? So the genetics of thermal tolerance is complex. Recall that most of these markers were found in non-coding genes. So areas of the genome that don't code for anything at all. We don't know what these are doing, even in well-studied organisms. We don't know what these are doing. So in non-model organisms, it's even more difficult to find this out. So while I think that these markers are very important and certainly biologically interesting, are we going to be able to have genetic assisted breeding and gene flow and selective breeding programs based on these genetics tomorrow? Absolutely not. So these genetic markers, you notice, vary between families, which means that they're likely to vary between populations, even more likely to vary between regions. So a lot more work would need to be done in this area. However, the take-home of this is this demonstrated heritability. So the fact that increased thermal tolerance can pretty reliably be passed from parent to offspring, and that we can assess adaptive potential in a population is very exciting, because these could identify populations that could be used for methods that Kate has proposed, Carly has proposed. Thank you very much.